Cath or Class Architecture Topology Homology. The Cath protein structure classification method is a semi-automatic hierarchical classification of protein domains. The database was first published in 1997 by Christine Orengo, Janet Thornton and their colleagues. Cath carries many broad features with its principal rival SCOP. Cath utilizes the classification of proteins as the basis of its organization. This classification is similar to enzyme classification that is used to classify enzymes. Like the EC system, Cath is numerically indexed hierarchy of protein domain structures. Cath cluster proteins at 4 major levels. Number 1, class. Class is derived from secondary structure contents of proteins. It is assigned for more than 90% of protein structures automatically. Cath recognizes 3 major classes, mainly alpha, beta, alpha-beta, which has both alternating alpha-beta structures and alpha plus beta structures, and a 4th class which contains protein domains which have low secondary structure content. Architecture. Architecture describes the gross orientation of secondary structures independent of connectivity in proteins. This category describes the overall shape of the domain structure. Topology. Topology of Cath clusters protein structures according to their topological connections and the numbers of secondary structures. This level of hierarchy is the fold level. Structures are grouped into fold families after considering the overall shape and connectivity of the secondary structures. It uses the SSAP algorithm, which compares protein structures based on the distance plot analysis. Only those structures that have an SSAP score of 60 to 80 are assigned to the same T level or fold family. Homologous superfamily. It groups proteins with highly similar structures and functions. The assignments of protein structures to the last 2 categories, that is topology and homologous superfamilies, are made by sequence and structure comparisons. Structures are clustered into the same homologous superfamily if they satisfy one of the following criteria. The first criteria is sequence identity greater than or equal to 35%, 60% of the larger structure equivalent to smaller. SSAP score should be greater than or equal to 80 and sequence identity should be greater than or equal to 20%. 60% of larger structure equivalent to smaller. SSAP score should be greater than or equal to 80, 60% of larger structure equivalent to smaller domains which have related functions. In Cath, the class level classification is done on the basis of the following 4 criteria: the secondary structure contents of proteins, the secondary structure contacts of proteins, the secondary structure alternation score, and the percentage of parallel strands in proteins. It currently stores 34,287 protein domain structures grouped into 13 superfamilies and 3,825 sequence families. In this image, you can see the results which are obtained when in the search box field text box hexokinase was entered. Cath data formats. Cath supports the following flat file formats: Cath domain description file. Information in this file format is compiled from the following files: Cath list, Cath domal, Cath domain FASTA sequences, Cath segment FASTA sequences, PDB-SU, file in CDDF format, and chain limits file in CDDF format. Cath domal. This file format describes domain boundaries for entries in the Cath database. Only PDB chains that have been split into domains have a Cath domal entry. Note that domains that are composed of whole PDB chains are not found in this file. Cath list. Cath list contains an entry for each structural entry in Cath. Representative structural domains are selected from Cath list based on the numbering scheme. Cath names. This file contains description of each node in the Cath hierarchy for class, architecture, topology, and homologous superfamily levels. It is organized into columns. Column 1 would have the node number, Column 2 would have the representative protein domain, which is a Cath 6-letter domain name. Sample Cath protein file. The structure record of the protein data bank is a unique 4-character alphanumeric code called PDBD or PDB code. Cath houses the same in its database, where information is described as follows. Title section. This section gives us information on the header, which is an ID code field which identifies the file from the other PDB files. Title denotes the title for the experiment or analysis. COMPND, which denotes the macromolecular contents. Source, this indicates the biological or chemical source. Keywords, the set of terms relevant to the entry. EXPDTA, an experiment information such as the technique used. Author, this denotes the name of the persons who submitted it. RevDat, the revision data since the release of the entry. JRNL, name of the featuring journal. Remark, any experimental details, annotations, comments. Primary structure section. This section gives us information about the primary sequence or the sequence of residues for each chain of the protein. This section also gives information about non-standard residues. Information about the name and formula of heterogroups in the macromolecule are also included in the section. SEQRES, sequence of residues for each chain of the protein. HET, non-standard residues such as prosthetic groups, inhibitors, solvents and ions. Secondary structure section. This section contains the data on the helices, sheets and turns found in protein. Position of turns, helices and sheets are provided, helix which are named and numbered. Connectivity annotation section. This section gives information about the existence and location of disulfide bonds and other linkages. Miscellaneous feature section. This section contains information that is unique to a structure, which includes special features like an active site. Crystallographic and coordinate transformation section. This section gives information about the geometry of the crystallographic experiment and the coordinate system transformations. Coordinate section. This section gives only information which pertains to the given atomic coordinates of the structure. Connectivity section. This section gives information on the chemical conductivity or how the atoms are connected to each other. Information here includes hydrogen bonds, salt bridges and links. Bookkeeping section. This section gives final information about the file itself. The Cath classification scheme is mostly automated, but some human intervention is required in the process. Various algorithms are used in Cath. Cath emphasizes on clustering proteins that have a similar fold so as to increase the understanding of sequence or structure relationships within protein fold families. Cath aims to improve homology modeling and structure prediction methods.